Hello everyone. In today's video, I will be describing a very important topic from the introductory part of autonomic nervous system, and that is cholinergic transmission or the cholinergic neurotransmission. Now, as the name suggests, the cholinergic transmission or neurotransmission involves a neurotransmitter named acetylcholine. So we will discuss the synthesis of acetylcholine, the storage, the release, and then the fate of the acetylcholine. I will try to explain the whole thing by the help of this pathway and then figure. Now this figure you can obtain from any of the standard pharmacology book, but the reference of my video is Goodman and Gilman. So it all starts from the synthesis. Synthesis includes the choline transport. We need to have a choline, and then choline is transported inside. Choline is transported inside. This is the nerve terminal, right? So choline is transported inside by a transporter named choline transporter, CHT1, choline transporter 1. Now there can be many transporter. There can be three important transporter, but here the transporter is of high affinity, which is named as choline transporter. Other you need not bother about. So choline transporter. Now choline is transported inside, and this step is very important for the synthesis of acetylcholine, and that's why it is named as a rate limiting step. Now where choline comes inside, there is acetylation of choline with acetyl-CoA by the help of an enzyme named as choline acetyl transferase. Before that, when choline is uptaken, there is the movement of sodium also. That is a symport. So choline acetyl transferase helps in the acetylation of choline with acetyl-CoA, and thus acetylcholine is formed. I will explain the whole process a little bit more. See what happens in the synthesis of acetylcholine. First, then choline acetyl transferase, this enzyme is synthesized in the perikaryon of the neurons. Now this choline acetyl transferase moves along with the exoplasm and reaches to the nerve terminal. And in the nerve terminal there is presence of mitochondria, and in these mitochondria there is acetyl-CoA synthesis. Now acetyl-CoA is synthesized here, choline acetyl transferase is synthesized here, and then it moves in here. In between the exoplasm is there, where there is choline is taken up by this transporter, choline transporter. Now three things which is mainly needed for the synthesis we get, and then finally there is synthesis of acetylcholine. Now the synthesis part is over. Second part is, after it is synthesized, what happens to that? It has to be stored. Where? Somewhere, and that is the vesicle. So it will be stored inside the vesicle. And how do they get there? Acetylcholine again is transported inside the vesicle by again a transporter known as vesicular acetylcholine transporter. Now this is transport acetylcholine inside. In lieu, there is a movement of H plus ion outside, and then acetylcholine gets inside. Now this acetylcholine is stored inside the vesicle along with ATP and VIP. VIP is Vasoactive intestinal polypeptide. Along with that, there can be calcium and magnesium and vasculine. They are termed as co-transporter or co-transmitter. Now, in the vesicular membrane, there are three proteins which are present. The black one is synaptobrevin, the red one is synaptotagmin, and the blue one is RAB3A or B. Now the significance of this I will tell a little later. Now it is stored inside. Now the storage of acetylcholine along with ATP is in the ratio of 11:1. 11 of acetylcholine and ATP of 1. Now there has to be release. After storage there has to be release. Upon depolarization of the nerve terminal, there is opening of a voltage gated calcium channel. We are discussing the release. Depolarization of nerve terminal, there is entry of calcium through this channel, which is known as voltage gated calcium channel, and this calcium goes and binds with these proteins. Now there are proteins which are present in this membrane also. They are, the black one is SNAP25, this blue one is MUNC18, and the red one is syntaxin 1. There are lots of abbreviation in there. I will give the details or the full form of the abbreviation in my description box. So this calcium comes and binds with here. And in the meantime, this vesicles moves from here to the active zone. Now in here also there are four phases. There is movement of this vesicle to active zone. There is the priming, there is the fusion, and then there is exocytosis. You need not know all those things. I just told you the name, the phases. So it moves from here to here near this membrane, and then this calcium associated complex with this protein binds with these protein, and then there is exocytosis occurs. That is the release of acetylcholine. There are two more important things. The first is depot pool and then reserve pool. There are two types of pool in here. Depot pool is there for a readily availability of acetylcholine, the release. Whereas the reserve pool, when there is prolonged depolarization, when there is a need of a release of acetylcholine for much longer time, then the reserve pool is used. Now acetylcholine is released. Now release, what happens? VAChT. Now we will discuss the VAChT. In VAChT, what happens is either this acetylcholine will go and bind to the receptors. It can be muscarinic receptor M1 to M5 or nicotinic receptor NN to NN. Now the structure of this is because it is a GPCR type of receptor. That's why the helical structure, or this is the ligand type of receptor. That's why this is named as drawn as like this. So it will go and bind here, or it will be degraded by the enzyme acetylcholinesterase to choline and acetate. This choline can be taken up. Or otherwise, it will go and bind to the presynaptic autoreceptor M1 and M2. There can be many muscarinic receptor depending upon the location. M2 receptor helps in the inhibition of the release. Now, so this is the VAChT. Either it will go and bind to the receptor, or it will be degraded, or it will bind to the presynaptic receptor. The hydrolysis or the degradation of acetylcholine by acetylcholinesterase takes lesser than millisecond also. Millisecond. Important thing to remember. Now this way the VAChT of acetylcholine happens. Now there are many steps, there are many parts where different drugs are used to increase the concentration of acetylcholine at the synapse or to inhibit it. What are those? We will see that. The first is this one. And the transport of choline inside can be inhibited by the drug hemicolinium. The second is this one. When there is the transport of acetylcholine in the vesicle, this can be inhibited by the drug Vasoactive. The third is fusion or the release. The release is inhibited, the fusion is inhibited by the drug botulinum toxin. And the fourth is inhibition of this acetylcholinesterase. So acetylcholinesterase inhibitor like neostigmine and carbamide. They inhibit this enzyme, thus inhibit the degradation of acetylcholine and causes more presence of acetylcholine at the synapse. So these are the four places where different group of drugs acts and inhibit or increase the concentration of acetylcholine. So this was all about the cholinergic neurotransmission. Hope this would be helpful. Thank you so much.